One of the more nuanced concepts in cellular biology that has practical nutritional implications is the idea that reactive oxygen species, the free radicals that antioxidants are designed to neutralize, are not uniformly harmful. This requires a significant mental adjustment for anyone who has absorbed decades of popular health messaging equating antioxidants with unqualified benefit and oxidative stress with unqualified harm. The reality is that the body uses reactive oxygen species as signaling molecules, and one of the most important cellular processes those signals trigger is autophagy, particularly mitophagy, the selective removal of dysfunctional mitochondria. The question for vitamin C and E, then, is not simply whether they reduce oxidative stress, but whether they do so in a way that preserves the beneficial RS signaling that keeps cellular cleanup running. This is where the synergy between vitamin C and E becomes scientifically interesting. Vitamin E, as a fat-soluble antioxidant embedded in cell membranes, primarily intercepts lipid peroxidation, the chain reaction by which free radicals propagate through membrane phospholipids, damaging cellular structure and potentially triggering pathological rather than beneficial autophagy. When vitamin E neutralizes a lipid radical, it is itself oxidized to a tocopherol radical, and this is where vitamin C enters the picture. Vitamin C, operating in the aqueous cellular environment, regenerates vitamin E from its spent form, a cooperative recycling relationship that extends the effective antioxidant capacity of both nutrients beyond what either provides in isolation. Together they function as a buffering system, reducing the excessive oxidative damage that could trigger pathological, damaging autophagy in healthy cells, while allowing the milder RS signaling that properly directs mitophagy to proceed. The practical food pairing logic that follows from this is straightforward and surprisingly appetizing. A spinach salad with fresh strawberries and a small handful of almonds combines vitamin K, vitamin C from the berries, and vitamin E from the almonds in a single dish. Bell pepper stir-fry with sunflower seeds achieves a comparable balance, bell peppers providing approximately 190 milligrams of vitamin C per cup, sunflower seeds approximately 7.4 milligrams of vitamin E per ounce. The fat from seeds also matters here because vitamin E requires dietary fat for absorption, which is why whole food sources that naturally co-package fat and vitamin E nuts, seeds, avocado tend to deliver better absorption than low-fat vitamin E supplements. The supplementation concern worth naming directly is the evidence around high-dose isolated alpha-tocopherol supplements. Several large randomized trials, including the alpha-tocopherol beta-carotene cancer prevention trial and the SELECT trial, found that high-dose supplemental vitamin E not only failed to prevent the diseases it was tested against, but was associated with adverse outcomes in some populations. This is not a contradiction of the food-based vitamin E evidence. It reflects the difference between a single isolated tocopherol form at pharmacological doses and the mixed tocopherol and tocotrienol family found in whole foods. The lesson is that the antioxidant balance that supports optimal autophagy is better maintained through dietary vitamin C and E than through aggressive supplementation with isolated forms that may disrupt the RS balance rather than preserve it.